Okay, so the main business of this morning is our keynote speaker. We're very lucky to have Dr. David Linden, professor of neuroscience from Johns Hopkins University, come to talk today. I've prepared a little bit of background on David. So David did his PhD with Arya Rautenberg at Northwestern University, particularly on the role of PKC and LTP. And then his postdoc was done studying cerebellar LTD with John Connor at Roche. And so he's really was one of the early investigators in the field of synaptic plasticity, particularly having studied LTP and LTD. It's a field that was always very contentious. So there were holy wars over presynaptic versus postsynaptic locus of LTP, LTD, and researchers could literally argue about how many angels can dance on the head of a spine. David, on the other hand, I felt was always kind of a voice of reason in this wild west. And he, I would say that in the holy war of LTP, LTB arguments, David has this kind of Talmudic logic almost. And so I hope, he's not going to talk about about his synaptic plasticity work today, but he's got a number of really exciting research interests, and I think he'll be focusing today on regeneration. David is at the Johns Hopkins School of Medicine, which he joined in 1992. As a faculty member, he was for many years the editor-in-chief of the Journal of Neurophysiology, and is a fellow of the American Association for the Advancement of Science since 2009, and a fellow of the American Physiological Society since 2015. In addition, David's received a number of awards, including a prestigious award from the Alfred P. Sloan Foundation for his public science writing. And if you ever have the opportunity to, if you want to enjoy reading about neuroscience in a leisurely and stimulating way that just makes you think and is very accessible, David has some fantastic books that I strongly recommend. New York Times best-selling books. Books on his first book, The Accidental Mind, had widespread critical acclaim. He also published The Compass of Pleasure, Touch, and most recently, Unique, The New Science of Human Individuality. And I just want to point out that David's books, as a testament of how popular and accessible they are, have been translated into a number of different languages. And I don't think I have the complete list, but I have Spanish, Italian, German, Japanese, Korean, Turkish, Chinese, Dutch, Russian, Portuguese, Thai, Romanian, Arabic, Slovenian, and for some reason, that may no longer be true, but as far as I can tell, his book about pleasure is the only one which has been translated into French. So David has done an enormous amount of eclectic research on topics ranging from synaptic plasticity, in vivo imaging, neuron-glia interactions, and I think today he's going to talk about axon regeneration. So David, I'll hand over the podium to you. I hope this works. Yes.
Let's go back. One. Okay, well, thanks, Ed, and thanks to all of you for being here today. Yeah, my book on pleasure was translated into French, and as Ed says, it was the only one. I was told by my literary agent that French people want to read books by French intellectuals, not English-speaking intellectuals, so that I would never get a book translated into French. So they did, but they gave it the most grotesquely unappealing cover, ensuring that no one would ever buy it. And the cover was a hamburger, but the buns were models of the brain. So there was half the brain on the top, and then lettuce, and then a meat patty, and then the other half of the brain. And it was the foulest thing you've ever seen, and so it's not surprising that that book didn't really sell very many copies in French at all. So today I'd like to tell you about one of the 5 projects going on in my lab, and this is about axon regeneration in the adult mammalian brain. And you might be thinking, well, but isn't that not a thing that happens? The dogma is that when axons are severed, either in traumatic brain injury or by drugs or in a stroke, that while they can grow back in the peripheral nervous system, and they can grow back in the young mammalian brain and the adult mammalian brain, that no longer happens. And what I would like to convince you of today is that there are a couple of cases where it does happen, and hopefully understanding those will allow us to develop therapies to promote recovery of function after brain injury more generally. So I'm used to using a pointer, but I don't think I can do two screens simultaneously. So I'm going to try to use the pointer. Is it visible? Yeah. Okay, all right. I'll try to use the pointer on the screen. So let's talk about our terms here. Regeneration doesn't just mean regrowth. Regeneration specifically means that if you have a severed axon, you've got growth, indicated here by this dotted line, from the stump of the severed axon itself. This should not be confused with local sprouting, which is a situation where you sever one axon and then its neighbor, in a compensatory way, sprouts a bit of axon out to innervate the target that was de-innervated when the axon you cut was damaged. Now, in the mammalian brain, in the adult mammalian brain, regeneration examples have been few and far between, whereas local sprouting has been well established as a mechanism going back to the 1970s, seminal work of Ozzie Stewart and others. So I would like to tell you about a system where axons can indeed regrow following damage. And this is of course a sagittal section of a rodent brain, and these structures here, the dorsal raphe and the median raphe, are part of the rostral group of raphe nuclei. There are also some caudal ones that innervate the cerebellum and the spinal cord. But the interesting thing is that these neurons project axons that pretty much go everywhere in the brain. They go to the cortex, the olfactory bulb, the thalamus, the hippocampus, and interestingly, to go to the cortex, they don't take a very direct route. So if you're a serotonin neuron on the dorsal raphe and you want to innervate the occipital cortex, you take this big C-shaped projection down through the medial forebrain bundle, ascending through the frontal cortex, through the parietal cortex, all the way to the occipital cortex. So these are actually some of the longest neuromodulatory axons that you will find in the brain. And they can be lesioned by amphetamines. And many of you have probably heard of damage to the serotonin system from methamphetamine or from MDMA. But there is one particular amphetamine called PCA, or parachloroamphetamine, here, which is the very most toxic amphetamine. And fortunately, as of yet, parachloroamphetamine is not a drug of abuse. I hope that no one is going to see this like if you put it up on the internet and decide, I'm going to go home to my lab and cook up some parachloroamphetamine and get really fucked up, because it would be, it's bad. So let's talk about parachloroamphetamine and how it works. It kills serotonin neurons at their release sites. So here, this is supposed to be a serotonergic synapse with serotonin-filled vesicles that fuse and release, a serotonin transporter that recycles serotonin back into the cytoplasm, and a vesicular monoamine transporter that packages serotonin into the vesicles. So PCA, shown here in red, is actually transferred through the serotonin transporter into the cytosol. It's then taken up by the vesicular monoamine transporter. It gets loaded up into the vesicle. Now, the vesicular monoamine transporter uses a proton gradient to power putting serotonin in the vesicles. The highly charged PCA molecule breaks down this charge gradient, and then this causes the VMAT to run in reverse, and so you get serotonin accumulating in the presynaptic cytosol. Then that causes the serotonin transporter to run in reverse, and you get lots of serotonin flooding out of the neuron, which is presumably what produces its stimulatory psychoactive effect. In this process, however, this is enormously energetically expensive, and that seems to be related to the fact that PCA kills serotonin axons by acting at their release site. So let me show you sort of the seminal finding here in this field that was done in the 1990s by the anatomist Mark Molliver at Johns Hopkins. And what he did is he made an antibody against serotonin itself, and then he had rats, and he would treat them with PCA, 2 doses in a day, and then he would sacrifice different rats at different periods of time after PCA treatment. And here you can see control. This is dense. The white axons here are the fluorescent serotonin axons. And you can see after PCA treatment, 4 hours later, basically they're completely gone. 3 days, they start to come back. 2 weeks more, 4 weeks more. And so Molliver's interpretation of this was, well, you got these axons, you kill them, they die back to the cell body, but you don't kill the cell body. And indeed, he did histology to show that you don't kill the cell bodies. And then slowly, over a period of weeks to months, these serotonin axons grow back through that C-shaped projection I showed you earlier, and then they regrow, and what you're seeing in this immunohistological picture is the serotonin axons regrowing. Now, how come this isn't in your textbook? How come everybody doesn't know about this? Well, the reason is that, well, there are 2 reasons. So this is Molliver's idea. These different amphetamines, PCA is one, here's some other ones, cause degeneration, the axon dies back, and then it regenerates and regrows. But people said, what if, because you're staining for serotonin itself, when you give the PCA and then you don't see any fluorescence, you think that the axons are dying, but what if the axons are there, they're just devoid of serotonin? Maybe the axons are just hollow and the serotonin is depleted, and then the serotonin refills the axons that were there all along. So that's one objection to this experiment. The other one is that when you do fixed tissue, you can't tell the difference between true regeneration, that is to say growth from a cut end, and local sprouting, that is to say growth from a spared axon that was left. And even though you can't see it that easily in this picture, there are actually some spared axons left if you zoom in this picture. So maybe they see the regrowth, and this isn't regeneration at all, it's just sprouting. So how do you address this? Well, of course, when Molliver did this, 2-photon microscopy hadn't been invented, and transgenic mice actually hadn't been invented either at that point. So now, with modern techniques, what we can do is we can make a back transgenic mouse using the serotonin transporter to drive the expression of soluble GFP and put it specifically as a specific marker of serotonin neurons, and then we can make a craniotomy overlying the neocortex in adult mice, and then actually do time-lapse imaging over months to even a year to see what's going on with these axons and make movies. So now, instead of doing a between-animal comparison, now we're doing a within-animal comparison, and we can see what's really going on. Is it sprouting? Is it really axon degeneration or not? Because we're not staining serotonin anymore, now we are imaging GFP. So this is just a picture showing validation of the mouse. These are coronal sections through the dorsal and the median raphe. This is the sort of butterfly or I guess dragonfly-looking structure here, and this is just showing that the GFP co-stains with serotonin or with tryptophan hydroxylase 2, which is the serotonin synthetic enzyme. Here in the cortex, the axons co-stain with either serotonin or with an antibody against the serotonin transporter itself. Just to be clear, in this mouse, we use the serotonin transporter promoter, but the protein that's expressed is not the promoter fused to the serotonin transporter fused to GFP. It is soluble GFP that's floating in the cytosol everywhere. All right, so it turns out that mice are much more resistant to amphetamine than rats because their enzymes that degrade it are much stronger. And so we have to do what we call the 4-day electronic dance music festival. And the reason we call it the Electronic Dance Music Festival is not because we actually play techno music for the mice. It's because it's a big problem. If you ever go to raves, you know that there's some people who take amphetamines and then they become hyperthermic and die. And that's why it's a good idea to go to the chill-out room if you've been dancing in a hot room with your friends and taking amphetamines, as people love to do at these festivals. So what we do is we give the mice 2 doses a day of parachloroamphetamine, the super toxic amphetamine, for 4 days, and then we actually blow fans on them to keep them from dying as a result. And so this is what it looks like in histology first. And basically, this is more or less the same result that Molliver found in his rats, standing for serotonin. So let's look here at the somatosensory cortex in a saline-injected control, and this is 1 week later after the end of the Electronic Dance Music Festival. And you can see the green axons are significantly depleted in the somatosensory cortex. They're depleted in the frontal cortex. They're depleted in the rostral medial forebrain bundle, shown here, number 5 on this trajectory, but not so much in the caudal medial forebrain bundle and not in the posterior hypothalamus at all. And so if you make a graph for this, here shown on the right, and what it is here's the x-axis is like the length of the axon, starting in the dorsal raphe, and then these are stations along that C-shaped projection. And what you find is that the initial part of the axon is not damaged 1 day after PCA, as shown here in the red circles. But then starting here in the medial forebrain bundle, you start to see damage until about 70% of the axons are dead in the rostral forebrain bundle and everywhere distal to that in this pathway. And then the other thing that's really interesting is that if you wait for recovery, this is 3 months in cyan and 6 months here in blue, you can see significant recovery of the GFP axon density during that time to prove that the cell bodies in the raphe are not themselves damaged. We did histology on them as well, and their density is normal. So this is consistent with a model that you give PCA, these axons in the cortex die back all the way to the base of the brain, down to the medial forebrain bundle, and then with time they grow back in. So if that's really true, we should be able to see it using a cranial window and 2-photon microscopy of the more superficial layers 1 and 2 of the neocortex, and we are looking in the somatosensory cortex. And so I think many of you do these kinds of experiments. Here's the objective. Here's the anesthetized mouse with a little isoflurane cone. Here's the cranial window, about 1 millimeter on a side with a piece of glass glued in the head bar to stabilize the mouse. And then here is the timeline. So we make the craniotomy. We give 21 days for the glial activation from the trauma of the craniotomy to cool down. Then we take one control image. We wait 3 days. We take another control image. We start the 4-day Electronic Dance Music Festival, and then we image the day after, and then every week for as long as we can keep the mouse alive and hold the window open, which typically is 3 to 6 months, but occasionally is as long as a year. And here's what these images look like. So these are 3D projections, and here you see dense green axons, but I also should like to point out, I don't know if you guys can see here, you see this kind of tube here formed of dots? It'll be easier to see when I rotate it. But this is not a neuronal structure. It turns out that the serotonin transporter is also in blood platelets. So you see blood vessels as these little dotted superhighways that run. And so keep your eye on the blood vessel here, and it's a little easier to see as I rotate, right? But the nice thing about these images is you can take them and then you can trace them in 3D space You can measure their varicosities. You can measure their relationship to each other. And at the time we first started doing these experiments a number of years ago, the postdoc who did this, named Yunju Jin, she had a small army of Hopkins undergraduates trace this. Nowadays, with AI, it turns out that it's dead simple. You can have the computer trace this in about 20 seconds, and then you only have to, once you've trained it well enough, and you only have to correct a few little errors that it makes. So the latest version, if you guys run Imaris, the latest version with the latest AI in it, it's the bomb. It's worth getting, and it will save you lots of money paid to undergraduates. I used to tell the undergraduates, I said, we're going to give you a trial on this tracing, and about half of you aren't going to be able to do it, and we'll fire you. And if that happens, don't feel bad, because like I can't do it. I don't have the, because it's a job that's both extremely boring and you also have to be very precise about it, and which is the worst kind of job in my view. And I said, if you can't do it, like don't feel bad. This doesn't bode well. You can be smart, you can be a good person, you can be caring, you can be good in your heart, you can be honest, and still not be able to do this job. I said, conversely, if you're really good at this job, that doesn't tell you anything about the rest of your life at all. Like, it doesn't mean anything else is going to go well for you. But we had a few people who were really good at it, and they traced, and here's what it looked like. So top here, you're seeing a top-down projected z-stack. So we've got a volume through the brain, where as if you were looking down from the top is what this projected z-stack is, and showing these axons. Again, you see right here, there's a blood vessel. Here's the blood vessel with the dots. So in here, this is a 3D projection of traced axons, and they're color-coded. And the blue-purple ones are the ones that are going to die when the PCA comes, and the pink ones are the ones that are going to survive. And then these colors are the newly added ones that regrow after the PCA. So if we look at the controls, the first thing to notice is that these axons are not really dynamic in the adult brain. They are very, very stable, not just in terms of their statistics overall, like their density, but they're actually very stable in terms of their position. They're very stable even in terms of their varicosities. If you go back 3 days apart, 95% of the varicosities that you see on this axon will be in the exact same place 3 days later as they were. So they're much more stable than, say, dendritic spines tend to be on a pyramidal neuron. All right, so here comes the Electronic Dance Music Festival, and you can see most of the axons are gone. The pink ones are left, and then you've got some new ones coming in, and we use a different color depending on the week they enter, and you can see the recovery of density, and you can see this recovery in total axonal length plotted here. Here's an example from a saline-treated control, and again, you can see that these pink axons, they're basically the same every time. So from minus 0.5 weeks to 15 weeks, it turns out that these axons, not only do they have the same density, but they're almost exactly in the same place. And over 15 weeks, you might have 90% conservation of the varicosity location as well. So the great thing about having these images is that we can look not just at total axon length, but we can ask some very particular questions. And that is, from the surviving axons, are we growing new sprouts? And is that contributing to the increase in total axon density? And if you look here in the orange triangles, you can see, okay, so here's total. You give the PCA down, you're down to 20%, and the total gradually comes back over many weeks. If you look at the sprouted axons, they're a very, very tiny contribution. As a matter of fact, if you compare it with the saline, the sprouting is at the same very low level in saline animals as it is in PCA-injured animals. Almost entirely, the new axons that are filling that volume are axons that are entering the volume from outside and are appearing new. They are regenerated, we believe, rather than sprouted axons. The other question that you can answer is, all right, well, you got these pink axons that survive here at day one. Maybe they're just dying slowly, right? Like maybe those axons really are going to die, but if you just wait a little longer? And the answer is no. Actually, if they've survived the PCA, those axons are going to then survive at the same rate as control axons. So there seems to be something special about them that is PCA resistant, and that's kind of interesting too. So when axons grow back, well, there's a big forest, I won't really say, but what we can say is that we can trace the varicosities along these axons in the in vivo images, and the density of the regrown axons is no different than the density of control axons or survived axons. And when axons grow back, they don't grow back exactly where any axon used to be. And so I think this is really important, because one possibility is that when an axon dies, it leaves a trail of breadcrumbs, so to speak, in the extracellular matrix, and then an axon comes back, maybe the same axon that was there before, or maybe a different one, and it follows that trail of breadcrumbs and regrows in that same channel. And that doesn't seem to be what happens at all. So the crossings of where axons used to be is entirely random. They're not following the paths of degenerated axons. In addition, when axons come back, they replicate the neighbor relations that they had that were there before lesion. So for example, layer 1 has a higher density of serotonin axons than layer 2 in the neocortex. When axons grow back, more grow back into layer 1 than grow back into layer 2 to reestablish that differential density and neighbor relations. Are these axons functional? Well, they can be stained for VMAT in the varicosities, so that's, you do EM pictures, there are vesicles in them, but do they really function? And so in order to do this, we collaborated with Perry Hashemi's lab, then at Wayne State. She's now at Imperial College in London. And Perry Hashemi is one of the few people in the world who can do voltammetry for serotonin. So I think many of you have heard of using voltammetry. It's a coded probe that you can use to detect the release of dopamine. Turns out the serotonin signal is 100-fold smaller than the dopamine signal. So it's extraordinarily difficult to do. And so if you give a burst of stimuli to the medial forebrain bundle and you look at the evoked serotonin voltammetric signal, here it is in a saline control, 1 week, 3 months, and 6 months. Here, 1 week after PCA, it's obliterated. 3 months, it's still obliterated. And it comes back at 6 months. So the problem with this is, in order to get the serotonin signal, you got to stimulate these axons faster and longer than they ever naturally fire in vivo. So yeah, so the voltammetric signal comes back as the axons regenerate. Is that really ironclad evidence? I don't think it is. I think we can do better. And in my mind, the two ways to do better, one of them would be to one of these new serotonin receptor hooked to GFP optical probes for serotonin, and that work is ongoing. The other way, which I think is really interesting, is that, so serotonin axons, as they grow through the neocortex, make two different kinds of synapses. 19 out of 20 synapses are what are called volume transmitting, and they're not real synapses at all. If you looked in the electron microscope, you'd see a presynaptic varicosity, and then you'd look on the other side for the postsynaptic density, and you would see bupkis, right? Nothing. There's nothing there. Because what happens is the serotonin is released, and then it just kind of, it's like throwing a bucket of water on the sidewalk. It just sploosh, goes everywhere, and it binds receptors at quite a distance after diffusion. 1 out of 20 synapses, which tends to be made on an interneuron, is actually a conventional synapse. And some of those synapses activate the one serotonin receptor, 5-HT3, that is ionotropic rather than metabotropic, and it produces a inward current if you're clamping at rest. So you can actually measure a 5-HT3 receptor-mediated inward current. So another way to know if these axons are functional when they grow back would be to have a 5-HT3 receptor promoter-driven fluorophore indicator mouse in a color that's different than your green serotonin axons, put them together, cut a brain slice, or if you're macho, pouch them in vivo, and then ask if you can get those currents back. So these are experiments that should be done. We're not going to do them, probably, but maybe you guys can do them. All right, so some initial conclusions. So do the serotonin fibers truly degenerate and regenerate as Moliver claimed but didn't prove? Yeah, they do. Is it compensatory sprouting that is responsible for the recovery? No, it isn't. Do the serotonin fibers that initially survive PCA treatment also survive long-term, or do they merely die slowly? They survive long-term. When the serotonin fibers enter the volume, do they enter as a bundle and then disperse, or do they enter individually? Well, they enter individually, poking in one at a time. Do they tend to grow along existing, surviving serotonin axons or blood vessels as a guide? No, they don't. What fraction of regenerated serotonin fibers survive long-term? And the answer is the regenerated ones survive normally, too. So when they grow back, it's not like they grow back temporarily and then die. They also survive normally as the spared ones survive normally. And as far as we can tell, the serotonin axons that grow back are normal, both in their morphology under the light microscope, their morphology in EM, which I didn't show you, and also this one suboptimal serotonin voltammetry experiment that I showed you. Now, is there anything special? The serotonin axons that survive, can we predict which ones that survive? Are they going to be the ones, we'll say, with the most neighbors or the fewest neighbors, or dense varicosities or less dense varicosities? No, there's no morphological signature that we could find that would predict which ones would survive. There's probably a gene expression signature if we could have RNA-seq for all of these, but we don't know what that is. Now, I'm not going to show you the data, but there are some behavioral deficits that are produced after this amphetamine treatment. One of them is in cued startle behavior, another is in the forced swim test, and those deficits do recover as the axons grow back. So we think they probably are functional. So here's the thing, right? We're looking through a little hole at the cortex, right? And the axons we think are dying back all the way to very far away to the base of the brain where we can't see through our window, and then we think they're growing back. We can't see that spot. So, we know that the recovery is not local sprouting, but is it really regeneration from the cut end, or is it distant sprouting? What if the sprout emerges outside of our field of view at the base of the brain, and then that axon grows in? That would look the same, wouldn't it? So what we did instead was something that I think is a very Baltimore approach. We stabbed our mice in the head. And so, no, literally, you take the scalpel and you stab the mouse in the head. We were kind of watching The Wire while we were thinking about these experiments. And so you can, what you do is you take the scalpel and you have the axis of the scalpel. If I'm the mouse, you put it in like this. So you're transecting the course of the serotonin axons as they're running from anterior to posterior in the brain, right? And then you make a coronal section. You can see the trench from the knife wound in the brain. And then you can stain. This is in histology. You can stain for the serotonin axons and show that, sure enough, after 10 weeks, they regrow. Here they are shown blown up. The dotted yellow lines are the trench. The green are the serotonin axons that completely grow across. And here we've traced a bunch of them in purple to show you, both up here in layer 1 and down here in layers 4 and 5. What about if you do this with in vivo imaging? So this is tracing from in vivo imaging. Look at all these cut ends that we made with our knife blade. And 9 weeks later, 31 out of 36 severed ends showed new growth when measured 18 weeks after injury. So we think regeneration from the cut end is genuinely what's happening here. So I mentioned earlier that the dominant mode of release of serotonin in the neocortex is volume transmission, right? A varicosity, a vesicle-laden varicosity with no postsynaptic specialization opposite it. So is that property of these axons related to their ability to regrow after amphetamine or stab injury? So one possibility, think about this. If you're a glutamate axon and you get cut and you regrow and you miss target, and instead of hitting where you used to be, you're now 200 microns away. Well, you may have done more harm than good. You know, you may have put the somatosensory map of the lip now in the place of the foot, or what have you, right? So conventional synaptic signaling has to be very precise to recapitulate function. However, volume signaling does not, right? Because you're releasing serotonin in a diffuse manner, all you have to do is get that axon back to the approximate neighborhood where it used to be, and you've recapitulated function. So maybe other axons that also use volume signaling can also regrow other neuromodulatory axons. And so we had a look at norepinephrine axons. Norepinephrine axons mostly come from the locus ceruleus in the brain. They also project very widely, interestingly not to the striatum, but to most other structures. And they can regrow following stab injury. So here is a mouse in which dopamine beta hydroxylase promoter is driving Cree, and this is a Flox GFP, crossed with a Flox GFP reporter mouse. And here's a stab, there's the trench, and here's the regrowing noradrenaline axons. Oh, so I should mention, this is, you can also use tyrosine hydroxylase. Now, tyrosine hydroxylase is going to stain both noradrenaline neurons and dopamine neurons. But the dopamine neurons project to the cortex, but only the deep layers in a mouse. They don't project to layer 1 and 2. So these are all, TH-stained axons are all noradrenaline axons here in this image. You can do the same thing. Instead of, with a chemical lesion, there's a compound called DSP4 that lesions the axons, but not the cell bodies of noradrenaline axons, in much the same way that perchloroamphetamine does for serotonin axons. And again, you can see the axon density come back slowly over time, over weeks, in a manner similar to what we saw in the serotonin axons. Now, it turns out that to answer this question of so first of all, right now, Patrick Cook in the lab is actively analyzing all the dynamics of the regrowth of noradrenaline axons with 2-photon microscopy, which he did. So we don't have it right now, but very soon we'll be able to tell you about the relative contributions of sprouting and the effects of on varicosities and so on. But what I can tell you about now is a very clever thing that he did. So, the problem is, what do you do if you want to measure the release of a neurotransmitter that only acts on metabotropic receptors and so doesn't cause ion flux in the postsynaptic cells directly, necessarily? You can't just put a patch electrode in the postsynaptic cell. And so there are different ways to do this. One of the ways is to exploit the fact that astrocytes are loaded with alpha-1 noradrenergic receptors, and that these receptors produce a strong calcium transient. This was discovered by Dwight Burgle's lab at Johns Hopkins, and has since been replicated by several other labs. And so here what you see in red, these are the living serotonin axons labeled with a DBH Cree crossed with a Flox TD tomato mouse from the Allen Institute. And then we've put GCAMP8 into the astrocytes with a viral injection using an astrocyte-directed miniature promoter shown here in green. So these green blobs are the astrocytes. The red is the noradrenaline axons, and these dark channels are the shadows cast by surface blood vessels that interfere with our imaging. So now what you can do is, remember before I was frustrated for the serotonin release experiments with voltammetry, because in order to get the signal, you had to stimulate those axons so hard you were in the non-physiological range. The great thing about this experiment, you do it in an alert mouse. You just give an air puff onto the neck of the mouse. That drives the locus coeruleus neurons very strongly, and then your GCAMP8 loaded astrocytes in the somatosensory cortex have a nice big calcium transient that you can measure, and you can block this entirely with an adrenergic alpha-1 antagonist drug called prazosin, right, to prove that it really is the noradrenaline release. And then you can ask the question in the same mouse, you can do this over and over before and at various points after DSP4, chemical lesion of noradrenaline axons. And you can see before the treatment here's a nice calcium transient in your astrocyte that's noradrenaline mediated. 6 days later in green, it's flat. 3 weeks later, it's there, but it's very, very slow. Well, why would it be slow? Well, keep in mind, this is the diffusion time you're seeing, right? So the chance of having an axon that's nearby your astrocyte of interest is low. So it releases over here, it diffuses, and then you get your calcium transient. And then, of course, as you recover, then you come back here. 9 weeks now, you're getting an earlier calcium transient. And again, at the end of the experiment, you can put the prazosin on again to convince yourself that it really is noradrenaline. So I'm going to leave you now at the end with some long-term questions. So is the unusual property of serotonin axon regeneration a general property of volume-transmitting axons? Well, maybe, because it's present in noradrenaline axons. We haven't tried it in other kinds of volume-transmitting axons. So we now know from expression profiling of serotonin neurons that there are 14 different types of serotonin neurons in the dorsal raphe. Some of them co-release glutamate and some co-release GABA. Are they all competent for regeneration, or is it just a subset? You know, maybe it's only one of the 14 types. Maybe it's all the GABA releasing or all the glutamate releasing, or maybe it doesn't matter. So this is going to be a fun thing to do, right? Because you can now, from Susan Demecky's work, using intersectional genetics, put markers in each one of these subtypes of serotonin axons. So, and then this is really the big question. What are the molecular properties of dorsal raphe serotonin neurons and locus coeruleus noradrenaline neurons that make them unusually successful at axon regeneration compared to most other neurons in the mammalian brain? So we, together with Susan Demecky, did some expression profiling of regenerating serotonin neurons and got a hit. We've yet to do the noradrenaline neurons. So we think it's going to be, having these two different pools, types of neuron that can both do it, is going to be really instructive in terms of limiting our candidate genes that might be involved in this process to what overlaps between the serotonin and the noradrenaline. And then finally, could we use insights derived from this to design therapies to cause other axons that don't normally regenerate, like GABA axons and glycine axons and glutamate axons, to do so? And if you could do that, would you want to do it, right? You might think, oh, well, that's going to be great. After someone has their TBI, I'll give this thing to regrow glutamate axons. Oh, shit, I just made them epileptic. Sorry. Right? Or maybe I made their neurological symptoms worse because the axons regrew, but they didn't regrow precisely enough, and everything is mismapped, and now you're worse than you would be. So it's not necessarily clear if, in the end, this approach would bear fruit clinically or not. All right, so I'd like to talk about the people who did the work, most of the work, the 2-photon imaging on the serotonin axons. was done by a former postdoc, Yunju Jin, and the work, that imaging with the stabs and with the noradrenaline axons was done by Sarah Doherty. Haley Janowitz worked on a project I didn't tell you about where we tried to use SSRIs to stimulate serotonin axon regeneration. It didn't work. And Patrick Cook has been doing the work on noradrenaline axons and the 2-photon imaging and the using the glial cells as an indicator of return noradrenaline release. And these are some of our collaborators. Chip Gerfen made the serotonin mouse. Misha Petnikov helped us with their behavior. Para Hashimi's lab did the serotonin voltammetry. And so as Ed alluded, I have a second job, and that second job is writing books about neuroscience for a popular audience. Here's the latest one called Unique, the New Science of Human Individuality, and I'm not going to talk about science communication and writing for a popular audience here at the podium today because I'm out of time. But if any of you are interested in that, please find me during coffee and cookie time or at a meal, and I would be happy to talk with you about that because I believe very, very strongly that it is to the advantage of working scientists that some of us pick up the mantle and communicate with the general audience directly rather than allowing science journalists to entirely do the job for us. No dig at science journalists who are, smart and hardworking people, but there's something about a lab scientist doing this that I think is beneficial, and I hope many of you will join in that endeavor. And thank you for your attention. I'm happy to take questions.
I'm not sure. Oh, yeah, it works. So thank you, David. That was really wonderful. If anyone has a question, just raise your hand and I'll come with a microphone to you. Here we go.
Hello, thank you. I was wondering, you were talking about serotonin receptors being on blood platelets. If you were aware of noradrenaline receptors also potentially being on these blood vessels. You were talking about, maybe the axon that died left crumbs behind to follow some kind of chemical gradient back to regeneration. I'm wondering if that might be true, but instead of the dead axon, like somehow the blood vessels are acting as this guiding cue for the regrowth of the axon itself. If there might be some interplay between regenerating axons and blood vessels.
Well, there doesn't seem to be anything obvious. I'm being gestured to go back to the microphone. There doesn't seem to be anything obvious in the sense that you don't see a blood vessel with axons growing along it or in parallel to it. The other thing is that you brought up noradrenaline. Now, the label that we're using for noradrenaline is DBH-CRE, a dopamine beta hydroxylase. So that's the synthetic enzyme for noradrenaline. You don't see that in the blood platelets. Now, it's possible, I don't know that much about platelets, and maybe there is noradrenaline in platelets, and it's not synthesized there, but it's taken up into them. I don't know the answer to that. I'm sure there's somebody here who does. But, so far, we don't have any indication of noradrenaline signaling for the platelets, and we don't have any indication of the blood vessels as providing a guiding role. And I should mention that we've done a lot of experiments where we visualize the blood vessels independently. So we have a mouse called Ty2-CRE MTMG that puts membrane-bound GFP in the endothelial cells of the blood vessels. We get beautiful images of the hollow tubes all throughout the brain. And when we label those with the serotonin axons, either in the regenerating or the control state, we don't see any particular association with them.
Fatima?
Hi. Hi. Thank you for your interesting talk. I was wondering, I kind of have two questions. The first one is, you mentioned that the new regeneration of axons are normal and they don't grow in the same place that the dead axon was. Do you have any evidence or thoughts as to if there's anything repelling the regeneration from entering that same space?
That's a really, really good question. We have done simulations, right, where we shoot axons with, we take the, what's the word for when things are twisted? Well, anyway, we take the average twistiness of the path of an axon, and we simulate those growing into a volume, and then we simulate where the axons used to be. And we ask, are they either actively attracted or repelled to that? It doesn't seem like they are. So there doesn't seem, in other words, they will readily cross those places where the axons used to be, but then they don't fall into the trough and grow along it. I would expect that if they were being repelled, you would actually see them turning away from where it used to be, and that doesn't seem to be what we find in the images. So I don't know that absolutely, but my guess is going to be there's not a repulsive cue left behind.
So potentially there's no limit to how many times it can regenerate. Like, let's say it regenerates and then you stab it again, or you give it PCA again. Will it regenerate again?
Oh, that's a great question. We haven't done that experiment. That actually turns out to be really, really important for low-level traumatic brain injury. And maybe this is the reason that you were thinking of asking it, right? So not the kind of, I got a car crash traumatic brain injury, but I am heading the soccer ball over and over kind of traumatic brain injury. And so I didn't show it here. Well, actually, maybe, oh, here it is. See this little divot in the brain here? So we do a model of a concussive traumatic brain injury, and the serotonin axons can grow around that just fine, the same way they do with a stab, CCI for controlled cranial impact. But we've never done a mild injury over and over again, like a heading the soccer ball model, to get at your good question. Yeah.
Cyril?
Yes, really interesting talk. I really enjoyed it. So I was wondering, today you were really showing data about axon regeneration. You said it's one of the 5 projects that you are studying in your lab. I was wondering if you're also interested in local sprouting. I was wondering, because I really found it interesting that axons regenerate like kind of the same conformation and like connections that they do before, as they were before. I was wondering if it's something that you expect to see also in local sprouting.
Yeah, well, so we thought we would see much more local sprouting than we did. Like, we thought we would see a bunch of local sprouting and a bunch of regeneration on top of it. And we were rather shocked when we basically saw no or almost no local sprouting at all in this case. So I don't know if that means that there's something about the presence of the regenerative program that is then disallows local sprouting. You know, that could be the case. Local sprouting, you know, a good model system for it, I may be telling you something you already know, but is in cross projections in the brain. So for example, the entorhinal cortex projects to the contralateral hippocampus and the ipsilateral hippocampus. If you lesion one entorhinal cortex and you look in the one that is denuded from that, then the projections from the ipsilateral entorhinal cortex into the dentate gyrus then sprout at the end to fill in. So this is one of Ozzie Stewart's models, and is a standard way of studying sprouting. Those are glutamatergic axons, and I suspect that the rules for these serotonin axons are somehow importantly different, but I don't have a guess of what that might be at a molecular basis.
Thank you.
Hi. Thank you for that really wonderful talk. It sounds like you have a fair bit of transcriptomics data that you don't want to, that isn't ready for prime time yet. But what I'm wondering is, from the functional analysis that you've done, can you comment on if you think this is an absence of of function? In other words, the cells are not responding in an inhibitory way to some inhibitory cue? Or do you think it's a gain of function where they have a mechanism that the glutamatergics don't have, and they can then grow?
Yeah, that's a really good question. So if I can rephrase your question, is when you're allowing these neurons to regrow, is it because they can't see the SOP signals or because they have a better motor? I think it's both. And the indications from the transcriptomic data that I'm not telling you are that it's both. But the one thing that I would like to show you is this micrograph, which I think is really amazing. So this is the neocortex laden with serotonin axons. Here's a stab right afterwards. 5 days after the stab, here the red is GFAP indicating activated astrocytes. Look at how these serotonin axons grow through this glial scar like it's not even there. So all the inhibitory signals from that glial scar that you would imagine would be turning away a glutamate axon or a GABA axon don't seem to be acting on the serotonin axon. And so, but that, I suspect that's only part of the story. It's not just a failure to transduce SOP signals. I think it's also, you gotta have a big motor. It's fine with me. I don't think the people on the web can hear you, but...
No.
Now starting to, yes.
Sorry about that.
Okay.
Wonderful talk. I'm really interested in the co-transmission of glutamate or GABA in the serotonin or dopaminergic neurons. When you're seeing your regeneration, you mentioned they're following a different path, but that doesn't really matter for serotonin because it's released in more of a volumic transmission. But when serotonin neurons are co-transmitting glutamate, I'm assuming the glutamate is more of a classical synaptic transmission with a presynaptic, postsynaptic. Is that true or is that not true?
No, that's not true, interestingly. And you're right that you would expect that it would be, right? So, no, if you look in the EM, opposite these serotonin varicosities, it's not like, oh, you see a PSD with glutamate receptors and no serotonin receptors. There's no PSD there at all. So presumably, the co-released glutamate or GABA is also in 19 out of 20 cases in the neocortex. And I should mention that these numbers are different in other parts of the brain. I'm just telling you about the neocortex, which is where we've quantified it. In 19 out of 20 cases, presumably that glutamate and GABA is volume transmitting as well. Now, I should mention that if you go back into the literature before your day, in the 1960s, some of it actually done by a famous Canadian neuroscientist named Krinievich, you find that there's evidence for glutamatergic volume transmission in the spinal cord. So it's not like volume transmission can't happen with these neurotransmitters that are most often transduced at a conventional synapse. They can. Exactly how that works, isn't very well understood. And how it works in the brain is really not understood at all.
That's really interesting. Yeah, I got to think about that and change my perspective then. I had a question about the stab injury. Like, how do you do that, but also image at the same time? Like, you're imaging right above the stab injury, or in a different region?
So, so for the stab injury, what you do is you can't have the way we do it is that we open up the cranial window. We don't have the glass in. We take an image, we stab, then we put the window on, right? So you're right, we can't stab through the window, although that would be a very Baltimore thing to do also.
Thanks for your talking. I have only simple question. Is the cognition of the animal is also recovered after the PCA treatment and the axon regeneration?
So is the cognition recovered? First of all, are there deficits and do they recover? And the answer is we did all kinds of tasks, behavioral tasks with this animal. We did learning tasks, we did a water maze, we did a Y maze, we did everything in the behavioral core, basically. The only two tasks for which we saw deficits after devastation of the serotonin axons with PCA, and I should mention that I'm showing you the neocortex, but actually PCA devastates all the serotonin axons in the entire brain, from the olfactory bulb to the cerebellum and everywhere in between. You would think that there would be more of a behavioral phenotype, but the only two phenotypes we saw were on forced swim test and on acoustic pre-pulse inhibition. We did see deficits with those, and those deficits did come back as the axons grew back. Now, you get into the weeds a little bit, I say as the axons grow back. Well, where do you mean? You're correlating it with, where we're looking is in the somatosensory cortex, which may not be the most relevant part. So do we know that the behavior came back at the precise time where the axons came back to the part of the brain that's most relevant for that task? That I don't know. But in a general sense, yeah, the behaviors that were impacted came back, roughly speaking, as the axons came back.
Hi, thank you. I have a kind of a two-parter. The first question is there any specific evidence that an axon that has now lost the ability to connect to its area, that it then regrows again to that specific area? And two, if it does, where do you think the signal comes from? Do you think it comes from the specific axon that is now degenerated is regenerating, or do you think it comes more from the area that has lost its connecting axons and then it's sending out a signal for that axon to kind of come back and follow?
Yeah, so that's a great question. So the first question is, when an axon dies back and regrows, does it actually grow back even to the general area where it used to be, or does it grow back randomly? We don't have a way of knowing that. Yeah, that would require some kind of complicated, interesting, multi-chromatic pre-labeling of those axons, and that's not an experiment that we've done. It is probably doable with some kind of rainbow animal, and that would be a really interesting thing to do. So your second part of your question is kind of presupposed on the first part, so I can't really answer that. Yeah.
Time for two more questions, and I will ask the last one. So there's one more over there.
So I had a question regarding the specificity. So if you were to train the animal to do a task, some sort of physical therapy sort of thing, would you expect to see accelerated regeneration?
Oh, so that's really interesting. So if we had an injured animal and we were doing some model of physical therapy, would that change the degree of serotonin axon regrowth? Yeah, so that's actually a really, really interesting question. Well, we haven't done the experiment. We've done something that's kind of tangentially related to that. So there have been a lot of claims in the rehabilitation after stroke literature that SSRI antidepressants can keep the window of effective rehab open longer and then allow for more effective rehab after stroke. Some people said, oh, well, that's just due to the antidepressive nature of the SSRIs. People are often depressed after stroke. When you relieve their depression, they're more motivated to participate in their rehab. And the answer is why. Other people said, no, you can give the SSRIs at a dose that's subthreshold for relieving the depression and still see effects on keeping the window open of rehab and having better ultimate outcomes. And so we did, rather by we, I mean Haley Janowitz in my lab, this experiment here, where she took a mouse, injured it with PCA, and then a few days after the injury, she started treating it daily with, this shows fluoxetine, but she also used sertraline at different doses to see if we could maybe make the regeneration of the serotonin axons proceed more quickly or with a higher varicosity density or with a different geometry, and the answer is no. Like, it was no different at all. So if SSRIs, this doesn't mean that SSRIs might not help with some plastic recovery process in some other kind of neuron or in some part of the brain that we're not looking at. But our hope was that we would see it here in neocortical serotonin axons, and we didn't. Yeah.
Okay, I lied about time for the last question. So actually, we're out of time. I want to thank David for a fantastic talk. Clearly, the audience was well engaged. Thanks so much.